Hello, everyone. This is Farming Fast and Slow. My name is Adam Wolf. And today's episode is called The Microbiome versus Everything, because I just have this experience that everything I thought I knew is suddenly irrelevant because of genomics and, next generation sequencing and, you know, there's a lot to be learned these days, and Adrian Ferrero is going to join us and explain it. Um, I Let me think a little bit of housekeeping as we get going. So the format is we usually talk about 30 minutes and have about 15 minutes for Q&A. Anytime a question bubbles up, feel free to post it, and we could start sprinkling those in, so don't feel like you have to wait for the invitation. These all get posted on YouTube. You can find the links to any of these on our website at arable.com/farming fast and slow. And, I was thinking about, kind of this episode. In some ways, we're so used to thinking about, in my little corner of the universe, about digital technology as being agricultural technology. But of course, genetics is kind of the most profound agricultural technology, starting with, crop breeding and, continuing to the present day, where we see, companies like Indigo, producing special coatings and whatnot. And so there's this whole layer of agricultural technology that is around kind of the informatics of how do you make sense of like unimaginably dense data. And I think, I first became aware of the microbiome thinking about, Sandor cats and wild fermentations, and like, gee, are we eating the foods that are allowing us to cultivate a healthy gut? And then I started my PhD 2005. This is at Stanford, and I met another student who their thesis topic was the microbiome of our teeth. I was like, whoa, this is something I never thought about, where there's, alpha diversity and beta diversity, where, the biome between one tooth and the neighboring tooth is like non-overlapping, so there's an ecosystem. Each of these is like a little, island. And another colleague was doing, I guess, fungal entophytes in Hawaii, in kind of natural communities. And then, I was aware that a lot of this is driven by the emergence of this 454 sequencer that suddenly allowed like a gigantic amount of data to be sequenced. And we saw, the Neanderthal genome sequence is like the marquee of that. And I didn't really track this. It's not my area of expertise until around 2014. We had this interesting paper come out in PNAS on kind of the biogeography of the vineyard microbiome. And this is by a student or a professor, pardon me, at UC Davis. And it totally, changed my perception of what we even think of as terroir in wine grapes. I'm somebody who really thinks a lot about microclimate, and to some extent genetics. And here there's somebody saying, no, it's neither of those things. And that's around the same time that I met you, Adrian, where you were actually turning this into just this immense array of insights around all different aspects of viticulture and enology. And, in that, I started to see, okay, there's this transition from understanding, genetics, genomics, to what I think of as biome science. And you have a company called Biome Makers. What is different about biomes than, just the genetics of a person like me?
So, hi Adam, hi everyone. So, pleasure of being here today. Well, you cover so many topics that are relevant
Sorry.
when it comes to microbiome, and especially when it comes to how to drive the curiosity around the microbiome and how microbes impact us. So, initially, when you think about this, everything initially comes to healthiness, but when we dive a little deeper, we realize that it's not just about diseases and health, but also about life itself, because most of the biological processes happening for living beings have happens because the microbes are around us. So in the skin, in the mouth, in the teeth, in everywhere. So they're all around. So when we realize about this and wanted to deliver value by using genetics to unveil this complex world and using computing science also to understand how these microbes impact us, that's the reason because we decided to travel from Spain to San Francisco back in 2015 and create biomakers to really start pushing harder on this new view, new approach, and understand really how the microbes, but not from the individual point of view, but as a
Sure.
community. You mentioned that we have a full ecosystem in our mouth. So
Yeah.
that's the reality. There is a complex community that is connected, and we have to really decode and find the way to understand this community and use it for our benefit. So that's what the reason because we decided to start biomakers.
And that's always, I guess, one of my kind of first points of entry. I'm somebody who's always trying to learn the names of plants, learn the names of birds and bugs and things, and I'm not very good at it. And in some ways, if you read natural history, people unpack so much knowledge once they're able to really identify something. Once you can name it, you can observe it, and once you can observe it, you can understand, well, why is this one different than that one, let's say functionally. And what I see is like, okay, well, what if I can identify, you'll have to correct me on the order of magnitude, a million, a billion different taxa in a sample, to turn that into a functional understanding of like good guys and bad guys, nitrifiers, denitrifiers. How do you connect, this snapshot of identity and turn it into a functional understanding?
Yeah. Well, it's not an easy task, and there's a lot of learnings and interactions just to be sure that you get to the end point. Let me just start from the beginning. If
Yeah,
you think about how we name the microbes, I mean, a dog is called dog. It's not Canis canis, or the wolf is not the Canis lupus, right? But
yeah,
microbes are always named by their scientific name. And if you think about, for instance, the Saccharomyces cerevisiae, which is, well, the microbe that we as humans have domesticated and
yeah,
use it for everything from beer, bread, so all kind of fermentative processes, still we call it Saccharomyces cerevisiae. So there is a lot of questions, a lot of unknowns and around the microbes. So if you see the initial reports that we were delivering, well, we started working in the wine industry because, well, we knew a lot about this
sure.
industry. Our families also produce wine in Spain. So we were aware of the relevance of the vineyard, the terroir, and how this can impact the fermentation process and the flavors in the wine. So that was like the perfect starting point for the whole development. But what we start doing is just delivering the list of microbes, and then realize that the value was zero, because you have a
Hmm.
huge amount of knowledge, and you mentioned it's really hard to retain that kind of information. And if you get a list of 700 microbes that are present into the soil, the grape, or the fermentative liquid, at the end, you don't know what you are getting. That's the reality.
Sure.
So that was the initial clue on how to move into more better interpretation of this community of microbes.
And that
So
hit you, I'm guessing, immediately as soon as you presented the first spreadsheet of like 700 species, that you have to start labeling them or annotating them in some way.
yeah, and exactly. And we were like species by species telling our clients what was the impact of this species. And eventually we just decide to turn the order, and instead of saying you have this bacteria, you have these species that are mobilizing nitrogen. And then we realized that we were losing a very relevant approach. If we wanted to use the microbiome as a biomarker of everything that is happening in biological processes,
Yeah.
the ecological component of this community. What are the interactions, the co-occurrences, the synergies between the different microbes? So we just hired a software engineer specialized in ecological computing and start developing models to really understand the complexity of this network. At the end, if you apply the same algorithms that the banks are using to understand the behavior of the people in the cities, which is in essence a network that is another community that is connected. So then you start understanding and make this interpretation, which are more relevant for farmers in this case.
Well, in some ways, I think that there's, for a farmer, it's very intuitive to think, you know, I'm growing a lot of plants, and my goal is to favor this set of plants over that set of plants. And once they overtop the weeds, then the weeds sort of can't compete. They don't see the light. And yet, when it comes to crop protection, we often think like, I want to nuke this population of let's say fungi, rather than manipulate the kind of population density of the good fungi versus the bad fungi, the good bacteria versus the bad bacteria, so that nature can take its course sort of in an ongoing way rather than a human manipulated way, you know.
Yeah. Well, the first thing is that we really like to see the microbes not as good and bad guys, okay? Because it depends on the environment. Let me just give you a very bad and stupid example. I mean, we can be very good person at professional level, but imagine you become crazy when you are looking at a basketball match, and then you start screaming, then you are a bad guy, right? So the same person, the same living being can be good or bad depending on the environment, okay? And this is important because even a pathogen can be positive if this pathogen is occupying an ecological niche and avoid other worse pathogens to enter into that ecological niche and protecting the plant. So it's better to be sick by a flu than be sick by a virus, no, Ebola or what other disease that is worse. Okay,
Sure,
so
sure.
this is a very good point to consider.
Yeah, I mean, that's very relevant now when we're thinking about flu versus COVID. Say, you know, let's say the immune response that you get from one bug could be leading to, you know, a wider scale immunity against these other bugs.
And it happens pretty much the same with plants. And there is another factor that is very important for growers. I mean, if you look back or, I mean, assess the current status of the soil fertility, actually, we haven't been very diligent caring for the soil fertility in the last 40 years. As a result, we have lost arable soil. And this is important. And now there is a lot of movements, a lot of awareness about regenerative agriculture, soil health, and so on. So we need to keep the soil alive because this is going to have a very positive effect for us as humans, for sure, because, well, carbon sequestration, biodiversity. Keep in mind that, well, the pandemic that we are suffering right now, probably if we will have a proper microbial biodiversity,
Mm-hmm.
that virus couldn't be around the world as it is so easily, right? And
Sure.
then plant growth, we need food to feed people, and we want to have qualitative food and quantitative food. And for that, if you can prevent diseases and can optimize the fertilization strategy, especially now that the use of bioactive products, biologicals, probiotic and prebiotic products are
Yeah.
becoming more normal, more usual in agriculture, well,
Sure.
we need these kind of tools to really match the needs and balance the soil functionality to ensure the productivity and the yield.
I I mean, what you're describing in some ways reminds me. I had a conversation earlier today about the power of the status quo, and we were talking about atmospheric water harvesting as it's kind of niche, but it's seen as niche because that's not what we do now. And in some ways, you're talking about biologicals as so novel, and yet we know that nutrient transformations in the soil involve all these same biological processes. It's just that the practice hasn't tended to employ these techniques, and so it's viewed as niche until it becomes the new status quo. Which
Well, well, what I really like to say when we talk to our clients is that, well, to provide them the possibility to look at their soil from a different approach, with a different angle, with different eyes, because we're not telling them which microbes are in the soil. I mean, that is available in our platform, so they can look for the whole spectrum of microbes that we have identified. But our report really contains very little information about the microbes. What it has is qualitative information and quantitative information about the different pathways that you can find. I mean, if you think about the use of nutrients, macronutrients as NPK, so
yeah,
you might be adding and spending money in NPKs, but the soil might be blocking those nutrients and taking them away from the plant. So the plant is not getting the source of the food to grow. Instead of that, you might need to add some biostimulant to really enable that pathway that might be blocked or be very active. If it is very active, you don't need to do anything. Just be sure that the level of nitrogen in the soil is the correct one, so the plant can be fed and not be stressed. But if you can also, for instance, another agronomic index that we deliver is related to the salinity, that is going to impact how the plant is going to grow, or the stress adaptation, to metal solubilization, or different hormones, or disease risk. So you can prevent before. So all this information is coming just from the observation of the microbial community. Of course, we need to digitalize the data, the biological data, and that's how we are using genetics. Genetics in agriculture has been used since many years because we have been breeding and we have doing experiments to get more stronger plants, let's say in that way. But now we can use these technologies as well to provide new data that can help us to do better. And that's what we are doing.
I mean, you have touched on, let's say, soil analysis, diseases, maintenance of biodiversity, crop health, crop protection. I know you've worked in, actual analysis of grape musts. How big is your scope? I'm always impressed at how many, how horizontal you've been able to be as a company and as an intellect. How am I catching everything? Are there other parts that you're tackling with biomakers?
Well, initially we started developing and proving the validation. So we were validating the technology in the wine industry. Then we realized that the real value today was in the soil, was for the vineyard. So we need the technology, the genetic technology to be improved, especially in the turnaround time, because if you have a problem in the cellar, you need a solution in minutes. Hours is too much. Okay, but when you talk about the vineyard, the soil, then you have a little bit more room. Okay, so everything is, let's say, slower than in the cellar. So then we also realized that if we focus our activity in the soil, we could expand to other crops. Okay, so that's what we started doing.
Like it's more generalizable.
1.5 years ago. Sorry?
It's more generalizable, the sort of insights that you develop.
Exactly. Yeah, because it can be applied, the same approach, the same tools can be applied to any crop. So right now we have around 45 active crops in our database. We have identified over 4 million taxonomic units, so different microbes. And we have processed 15,000 samples from 40 different crops. So and coming from different countries, different regions in the world. US is the main source of data, but we have samples from Europe, Latin America, Africa, Oceania, and also some samples from Asia. So let's say the way we understand and the way we aggregate the data to make this functional interpretation of the soil is very good, because it doesn't matter much where or which kind of block you are analyzing on the same crop, because when you look at the functional profile of that soil, there are not so many differences. If you compare the rainforest from Brazil and the rainforest in Australia, from the taxonomic point of view, is completely different. But from the functional point of view, the biological processes happening in the rainforest are pretty much the same. Of course, there is some environmental factors, elements impacting. Sure. But at the end, if you want to optimize that ecosystem, you have to look at the functional level. So that's what we have done. Today, we have more clients in Midwest from corn and soy than vineyards from Napa, but still we have quite many.
I mean, it's so, I think of this as macrobiology or macroecology, where we have redwoods in California, but in Australia, they have eucalyptus, you know, and in some ways they carry a similar kind of functional perspective. Or in high latitude North America, we've got, you know, all different parts of the Pinus family. But then if we go into South America, we've got like the Araucaria, you know, so functionally we've got things that fill the same niches. And I never had really, you know, connected that with the soil, where we're used to thinking, gosh, you know, tropical soils are just so different from temperate soils and Mediterranean soils. And what you're saying is actually, from a functional demands of a crop, a healthy crop, soils can be quite similar.
Yeah, it can be comparable, but of course, you want to compare as close as possible. Today, according to the data that we have, for instance, when we talk about vineyards or almonds, so crops that are very concentrated in California, we can give detail at California at state level. Okay, when it comes to corn, for instance, we compare at wider level, because that is what makes sense. So that aggregation is important, and also we'll keep improving the references, because the good thing is that our system learns. Today we are helping or training the system to make this interpretation, but as we feed the system with new data, the results become more robust.
In some ways, you know, more data gives you different analyses, because you can contextualize this sample based on, you know, nearby samples, previous years. Do you find that the arc of your analytics changes in light of having more data to work with?
It changes, they say it becomes more precise.
Uh-huh.
It doesn't change much. So where we were able to say like, this is a low value,
Yeah.
then we might say this is a very low value, okay? Because the way we deliver is from 0 to 5, being
Yeah.
0 like very low, normal, high, very high. So those are like the parameters. When our clients need further detail, there is some numbers behind and we can give up to 2 decimals. So yeah, but no, the reality is that you narrow down, okay, those references so you can better target the outcome. But no, we don't see like critical changes. Of course, anytime we launch a new crop, we make sure that we have enough references, because otherwise we'll not be able. And having enough references is something that we do by working with researchers. You mentioned at the beginning, UC Davis. We have made collaboration, I will say, more than 40 different researching groups all across the world. And the good thing is that they publish the data, so those data are accessible, and they are very happy if we help them to recompute the data with our approach.
Do I think of these, you know, we often have this kind of phenomenon where we originate data. Customers, you know, have data. They tell us, you know, I'm growing grapes or corn or what have you. But then there's this outside information that we need to get from the universities or sort of pre-existing research. It could be like turning growing degree days into growth stages, or, you know, it's just sort of outside context. And it strikes me that you have something similar, which is, you know, you must depend on other people to be identifying any particular named taxon to saying, oh, this is a nitrifier, or this, you know, what function it has. Are you ultimately limited by, you know, these outside labeling efforts that are
At the beginning,
describing?
yeah, at the beginning, yes. I mean, there is 3 international databases where researchers all around the world are feeding with the species that they discover. So this is an input that we use in our system, but still, this is open information. So you can imagine the amount of data, right, wrong, or not accurate that you can find. So what we did at the beginning, we spent a lot of time just creating the data, creating our own references according to what humans know. So
Sure.
what we have realized is that we are able to name, not to identify, because we identify everything. We're looking at the DNA, so we see everything that is there. But
Yeah.
from the whole microbiome that we identified, we're able to name around 40%, okay? 30 to 40%. That's the
Okay.
reality. The rest, it doesn't mean that it doesn't count. It means
Yeah.
that we don't have a name, so we just put a label like a number, okay? And then by looking at the genome, we're able to guess what is their metabolic potential because of similarity. We might not know the species level, but if we go higher on the taxonomic level, we make
Yeah.
association. And
Uh-huh.
then you have basic functions that you can profile and assign to those species, identify the same in different soils. So then you start understanding what are the ecological dynamics of those species. At the end, what we decide is to use the soil microbiome as a biomarker because, well, it has an active role in all the biological processes. It's a dynamic community, so it is impacted by anything that is happening in the field. So it's the perfect biomarker, right? And also has a role, a function, so something to do with plant growth. So what we do then is to estimate the genetic potential, the functions based in the genetic potential, or the functions based in the potential of the genes that we see in that community.
Sure.
Okay, and all of this is just mass computing, and that's how we are able to make this complete overview.
I mean, it's just an extraordinary amount of connecting the dots.
Too many,
Of course,
right?
well, and there's parts of it of, just seeing, let's say you identified 30% of it, that it strikes me that it is like a land grab might not be the right, but there's just everything you discover is new, and so there's just immense amount of like, gosh, I didn't make a company so that we could discover this, but it turns out that we are good at this, and suddenly new avenues present themselves.
Yeah, yeah. Well, soil is the most biodiverse environment that you can find. So that's the reason, because we identify the problem and the reason. Well, the problem first is that we are only able to isolate and grow, cultivate around 1% or less of the microbial species. So even if you know that is there, there is no way to really isolate and grow and use it for our benefit. So there, and that's the reason, because there are so many ag input startups or startups trying to develop new biologicals
Yeah,
by different techniques, also genetics as well, because they need to profile and then try to really isolate and grow. That's the real challenge, and then demonstrate what is the effect. In our case, as biomakers, we decide just to be the independent company providing the data, delivering the data. Our goal is not to deliver new biologicals. I mean, we work with input manufacturers, and some of them are very big, some of them are very small. They have very interesting products, and the goal at the end is really be able to give some light to open the dark box, the black box
yeah.
of the effects, because there are so many biologicals that even the growers tell us there is nothing happening here. This is my guess. I'm spending this money, and
Yeah.
I don't know if I got this yield because the product or because the nature itself. You know what I mean? So we
Well,
want to bring the light.
I mean, I grew up, with Grimm's fairy tales and Jack and the Beanstalk, and he sold the cow for some magic beans, and his mom is like, ah, you idiot! Like, what did you do buying magic beans? And I think, anybody that I'm talked to, you know, may well think I'm trying to sell magic beans, and let's say in the biological space, be it, compost teas or various, magic potions, it's hard to interpret the claims, but the work that you're doing, identifying, what is this community, how might it interact with the resident community in your soil, that seems closer to a predictive science, you know uh,
There is a lot. Yeah, at the end, what we are able to, and this is part of the R&D that we're doing, is is identifying the patterns and modeling predictions on what's going to be the yield identified. Yeah, this is kind of to deliver back the uncertainty more certain,
I
so we can navigate in better way.
I wonder, I mean, it's sort of maybe one of these holy grail questions of, how close are we to being able to design a system? And in some ways, I think our pea brains are too, small to be able to design a system that contains all of the essential complexity, because there's so many interlocking, interacting pieces. Um, are you making baby steps towards sort of making kind of prescriptions of, what do you need more of, what pathways are you missing,
Yeah,
can we
some
add some of
people,
those pathways?
yeah, some people is telling us, are you going to be the agronomist, the digital agronomist? Well, the reality is that agriculture is the oldest activity that humans have been doing. So there is a lot of empirical knowledge that we have, and we shouldn't lose that and lose that. And probably it's rather than difficult that a machine can replace everything, because at the end we are able to process, I mean, as humans, much
Yeah.
more complexity without less data, and then take decisions. The way we see it is that we should be suggesting, making the one like narrowing down what are the most suitable options, okay?
Sure.
And then the agronomists should be the ones really making the final decision. But the goal for us is to be the tool that will make their work easier. I mean, otherwise, you mentioned the claims. There is no way to really know what are the functional claims from, I mean, to verify. What, if
Yeah.
you host a dealer, they will tell you amazing things about their products. But at the end of the day, you really don't know what is the reality behind that product. So you have to trust a technology that is independent and can do this kind of assessment and provide the functional claims on the products. This is something we're really working on trying to create in for instance, the CDFA is already accepting the results that we generate on the experiments on the products, okay, to really accept functional claims in the labels, which is very important for us. And the same, well, Asia, India, for instance, Europe is now launching new legislation on how to label biologicals. US is also preparing this. So yeah, this is needed, and this is going to come some way or other. To be the solution for the complex system is too complex. Baby steps, I love that. Yeah, we're doing the baby steps, but I don't think it will be in our life the
Well,
ones doing that.
I love the way that you created it. It's almost like an expert flow chart. You know, so I was in a doctor's office. This must have been pre-COVID. So, you know, we did a blood panel, and you know, is gosh, your initial observation was you're anemic. We need to do some more tests, and it wasn't that as anemic for low iron, but for some other deficiency. Actually, what you need is vitamin D. And being able to have a flow chart of saying, okay, well, I have this first observation. We need to find out another piece of information before we know what's the appropriate treatment. Otherwise I'm taking iron pills or something. And what you're describing is almost, I want to say, like a blood test, which is, it's complex. It requires interpretation by an expert, but it can route you to, let's say, away from inappropriate paths of treatment.
Yeah, and we should think about that example is very good, because at the end, even if you have so many data around your body, I mean, you can monitor everything that is happening. When from time to time you go to the doctor, and the doctor will at your face and find you have this straight. No, you need to do this or this other. But having more reliable and accurate data is very useful, especially in healthiness. When it comes to plants, it's pretty much the same. We are feeding the plants, we are caring for the plants, and we are treating the plants against diseases. So at the end, what we want is to have food, the plant itself, of the products, the crop that the plant does. So
Yeah.
pretty much the same. But you need a doctor, that's clear.
Well, what I love is that it sort of takes, I guess, a technological opportunity, which is, I have a way to give an X-ray into the soil and give a community composition, and I'm going to have a bunch of metrics that, like a blood test, someday we might expect agronomists to be able to identify, everything and say, you're high, you're low, okay. And what that leads naturally into is like the product of how to benefit from the technology, which is, in some ways totally different from the technology itself. How do people benefit from it is through kind of what you've been designing, is kind of report cards and things.
Yeah, and today most of the agronomists lack on tools, and they are really guessing what to do. What we want them to do is to know what to do because they have data supporting their decisions. And that's the reason because we're working with PCAs, CCAs, because we want them not to be afraid of this unknown, okay? And it's something they can learn very quick, and we're happy to share everything that we have learned with, we have 350 clients, so we have quite good experiences.
Yeah.
And but still there is a lot of learnings to be done, and hopefully we can do it as a community as well. I mean, we cannot change the world by ourselves. We have to really team up and do it all together. Otherwise, this is not going to work. We can be the ones delivering some value with a specific tool, but we need others to really use the tool, combining with the rest of the knowledge, other companies delivering new biologicals, you know, this huge loop. And at the end of the day, what we're going to have is more living soil, soil that is more alive and is going to bring, you know, all the positive side effects that we need to be sure
Yeah.
that we are able to sustain the life and we are able to really have food for everybody. And there is another important element. If you look at the financials of the farmers and the yield growth in the last 10 years, well, you see that the average income hasn't changed much while the yield has increased. So probably there is some adjustments if we want the farmers to really benefit from the productivity, because there is a cost and we don't want the soil to be the one bearing the cost of this productivity increase.
Sure. Sure. Well, that won't end well. Um,
Exactly.
I have a question from our audience, which is when you assess soil pathways, what are some of the more common pathways that are missing? Put another way, if you were designing an intervention to help farmers, what intervention would you build? It's a great question.
Yeah, usually the way we do it is by the question they want to answer. Okay,
Mhm.
so the most common questions that we face is I have two different areas in my field, and the yield is different, and I'm treating it in the same way. So there is only one way to explain it through the biology of the soil.
Uh-huh.
So we see the differences, and then we see how we should balance one of the areas to increase the productivity, the yield, right? Another question that we face is we are transitioning to this
But
new
just
farm.
for the first case, you could test each of these and compare, how are these different? And the deficient soil is apparently, you know, uh, missing some of the things of the healthy soil. Is it is easy as subtraction?
Yeah, yeah, actually, because you have another data that is the yield. So you know the yield in this area and the yield in this other. So you assume that this soil profile, functional profile, is going to provide this yield, right? And this other is providing a lower yield. So then you see differences. It might be a nutrient block, it might be a disease, or it might be some plant growth factor. So
And
another,
and I cut you off. Yeah, what's the next most
yeah,
popular?
the other two, let me just go with two additional questions, is I'm starting to use these products. I want to see, to monitor if my soil is really benefiting from it. Okay, I'm transitioning to this. And this is very common, people moving from more like strict conventional practices into a mix or biological or, you know, organic farming practices or
Yeah,
whatever they are
yeah.
testing. And then the diseases also. Diseases is not an easy topic. So if we are able to see any threat related to infectious diseases, that's also a very common. I have this problem and I
Yeah.
want to prevent, or I'm going to decide which is my crop protection strategy. Can you tell me what are my risks? So we are going to tell them. Those are like the three most common questions.
One of the interesting, discoveries when I was first doing customer discovery before starting Arable was, I was trying to understand, who is my demographic? Is that defined by, people who are digitally astute? Do they grow certain crops? Are they a certain age? And one of the findings was, people who are interested in progressive practice, it could be no-till, it could be cover crop, they're interested in data in a way that people who are kind of following an existing program aren't. And so there's this phenomenon that's sort of correlated. You might not immediately think, people who are interested in no-till are interested in genetics, but suddenly you find out, gosh, there's a lot of people embarking on an experiment now, and that's what your target is, which is, I think it's kind of fascinating to see, where is, new technology being adopted.
Exactly, and you need data. I mean, we are in the society of data in our daily life. Beyond our professional activity, we use data. And what is more important, we are used to get the data right now and as much data as we demand. So if you have a question, now
Yeah.
you don't go to the library and look for the answer. You just go search it in the browser and find the answer. So yeah, I fully agree. And this is the way it has to be. And it has to be also easy and funny. This is important. That's the reason, because for instance, we have worked a lot on the visuals, how you play with
Yeah.
the results, because it's not just having the results, it's also having fun. So gamification is not
Yeah.
itself the proper word, but you try to deliver good experiences, because otherwise, where is
I
the
mean,
fun,
when I
right?
saw your materials specifically a few years ago at United Wine and Grape, I brought it back and showed my team is like, it's so excellent in terms of the presentation of the data, and the presentation helps to get the message that comes out of it. And the message can be quite subtle and brainy, and yet the presentation makes it relatable and digestible. And I think like it is an essential piece that is often, for scientists turned entrepreneurs, it's hard to understand why someone might not interpret a graph the same way we did, you know.
Yeah.
um and so it's I love I don't know how you've achieved such strong design even at the same time as having strong kind of fundamental, tech. We only have a couple minutes. And I guess, we've covered a lot of things. Is there any kind of frontier kind of directions or new ideas that you want to highlight before we go? What are we going to be talking about next year?
Well, hopefully we keep talking. That's the first question, no? Because when you develop a new technology and you bring the science into a product that you can touch, that you can use, that you have validated, there is success cases and so on, it's always important to create curiosity in the people. So my message is make people not feel afraid of these new products. It's a digital product at the end. No? It's a service, testing service. So you just send soil. You can send the soil to the same lab because we are building partnerships all across US. And you can also mix the physicochemical soil test with the functional test. So you have the whole picture, and
Sure.
you can connect, you can plug this layer of data into your precision ag platform, because that's also important. You look at the soil, but then you want to look at the water and all the parameters as the ones that you are delivering, and then you have the whole picture. And this is important. So that kind of support and curiosity, we are happy to interact with the people, teach, share knowledge. So happy to welcome anybody that has curiosity about the impact of the microbes in their crops.
Yeah, I mean, you're giving me so much food for thought right now. I almost need to pause so I can digest it all. Um, I really appreciate your joining us. I know your schedule is tight, and so I'm very grateful. Thank you, Adrian, for joining us.
Thank you, Alan. It's such a pleasure conversation. I really enjoy this session, and hopefully it's useful. So let's keep doing this because it's very useful.
And just so that people know, what's your website?
www.biomemakers.com, or they can check the product brand, which is Big Crop, BECROP, Big Crop.
bcrop.com. Okay. I'm excited. We'll obviously put those in the links. Thank you, Adrian. Thank you, Masha Crichton, our producer. And we'll see you next week. So thank you.
See you. Thanks. Bye.